Louise Myers, and today at our Team Journal Club, I'll be presenting a paper, ApoE genotype determines cell type specific pathological landscapes of Alzheimer's disease. And this is a recent publication by Lee et al. In Neuron. So the experimental design and background of the study is they're studying changes in transcriptomic and also protein expression in the human temporal cortex across different ApoE genotypes. So across the ApoE genotypes, there is increased risk with E2, with the E2 genotype having a protective effect, E3 being neutral, and E4 being the most risk for AD. So that's their axis there. They also have both male and female donors relatively evenly split for overall 29 AD and 27 control donors. And when they're performing single nucleus RNA-seq over this, yielding 343,000 nuclei over 7 major cell types. Looking at the single nucleus data set, they found that the Alzheimer's samples had lower counts and detected features, but had similar mitochondrial and number of nuclei overall. And then the proportion differences, they observed slight proportion differences with high proportions of oligo in 80 versus control. Microglia, they had higher proportions of microglia and ApoE versus ApoE3, and OPCs, AD had fewer OPCs than control for ApoE4. So even at the proportion level, there's overall differences like an oligo, but things start to modulate at the genotype level. And then digging into their kind of like, they did a big major differential expression analysis that then led to further refinements that we'll see in other figures and analyses in this paper, including some validation assays. But basically what they did is within each genotype and each cell type, they ran Alzheimer's versus control differential expression. Their modeling also included random sequencing saturation as confounding variables. And the first thing that they point out is that there's very different results between genotypes. So looking at the Z-score, oh wait, no, I did not zoom in on there. But I guess each of these little plots is a cell type, and then they're looking at the Z-score between E2 and E3. And if this change was like similar overall, you'd expect that correlation to have like a high positive correlation, but we actually see is like a weak negative correlation. That's kind of what we see for most of these cell types, except for microglia, where the stars here denote if it is a significant correlation. We can see that these are like inhibitory is like significantly negative. So I think that like that's maybe not what I would have anticipated, but they found like pretty different signal between the genotypes within these cell types. I think specifically pointing out that oligo E2 versus E4 is one of the only significantly positive correlations. On top of those sets of differentially expressed genes, they perform gene set enrichment. So for each of these little, they're not really volcano plots, but they're like comparing the normalized enrichment scores on the x-axis to the negative log 10 p-values. We'll zoom in on the excitatory one here, and they point out like some of like the most significant gene sets here. And then these are also color-coded by the genotype where the gene set is significant in. So here we see that like vesicle-mediated transport we see that is like downregulated in E4. You can kind of see those patterns where we see things upregulated a lot in E2, and then we see some downregulation in E4, E3 kind of being split. I'll also point out so excitatory and microglia are two cell types that they're going to zoom in on. So I just wanted to point these ones out. So yeah, that's synaptic compartments here, and then also for microglia, a lot of immune response. They pull out these enrichments. Yeah, so for the excitatory neurons, again, zooming in, we see an ApoE4 downregulation of synaptic vesicle cycle, other vesicle-mediated transports, and some of these genes involved in that. For ApoE2, we see the opposite, where we see an upregulation of synaptic transmission. So they kind of like pull this out as like a contrast between the two genotypes here. And then in ApoE3, there was like three only genes. I guess they're pulling out here where it's like these are E4 downregulated unique genes, E2 upregulated unique genes. These are their enrichments. For ApoE3, I think they had no like significant unique pathways, but they did found downregulation of some related synapse genes. So kind of like a similar theme there. Digging farther into the excitatory neurons, they did subclustering So they pull out 14 subpopulations and they annotate these by layer. And what they found is overall, they found similar compositions at the subtype level between AD and control. And they didn't really find anything, they just know a consistent enrichment of gene sets across subclusters. So they didn't really know anything where like one of these subclusters is super involved in that AD control difference. They didn't find anything too interesting there. But they then kind of zoom in on that synaptic protein loss, like what's going on there. So if you remember, we saw down synaptic gene sets in ApoE4 and upregulation in E2. So what they do is they're looking for VGLUT1 and PSD95, which are genes involved, or I guess they're looking for the proteins rather than the genes or transcripts here in the tissue. So here we're observing, so they observe the synaptic transcript changes, and they're saying, does this actually result in like protein loss in the brain? So we're using immunohistostaining and co-immunofluorescence to like check if this is supported. Wait, I think I had a animation here that didn't. Yeah, okay. So what they saw is like here we're looking for immunohistostaining of VGLUT1, and you can see between control and AD. So for E2, we see that these look the same. This is preserved. For E3, we can see that this tissue gets lighter. We see a reduction in VGLUT1 and AD. And then in E4, there isn't really a change, but you can see that overall it is lighter. Like for the control, it was lighter than E2 or E4. So that's like the change they record there. Um, and then they also do the co-immunofluorescence where they're showing VGLUT1 and PSD95, and they were gonna like they see similar changes. So like VGLUT1 is preserved, we see the decrease, and then you see like a consistent decrease over here. So overall, they conclude that ApoE modifies the synaptic changes in AD, preservation in ApoE3, overall reduction in ApoE4, but then like, or wait, reduced with ApoE3, that's the typo there, unchanged proteins in ApoE4, but like it's transcriptionally like downregulated. So this was, wait, sorry. They saw the reduction in ApoE4 at the transcript level, but then the proteins is unchanged. So there is probably something else that's like modulate, like between the transcription of the I guess like between there's like another complicated change here because we see this like mismatch between our RNA and proteins at the E4 level. So then another group of changes that they focus on is in oligodendrocytes. They're looking at changes in myelination. So they see that myelination is like commonly brought is like one of some of these like highest gene set enrichment results. And also, like we know myelin loss is a symptom of Alzheimer's. Yeah, so then the transcriptomic gene set enrichment showed upregulation of myelination in ApoE2. So they dig into this again. So again, we can kind of see this. They focus on kind of like these unique genes that are like only upregulated E2. Let's check out what those gene sets are. And we can see that there's like myelination, axon sheathment. For every three unique genes, we see a downregulation of myelination genes and axon sheathment. So kind of the opposite of this. And then including MVP, which is myelination basic protein. ApoE4, we see an upregulation of CNP. So there is less signal for ApoE4 here. As you can see that the sets of unique ApoE, the E4 unique genes is much smaller, but like it includes CNP, which is a gene involved in myelination as well. So again, they break our oligos into subclusters. They do find, they said there were similar changes across the subclusters, but they do note that E2 changes were more enriched in these populations, and E4 more enriched in those in 1 and 4. So there's some changes here, but they didn't find anything like super distinct. And then to validate their MVP changes, they first looked at their normalized expression levels of MVP in their single nucleus groups, and they note that MVP is lower in E3, E3AD, and in their oligodendrocytes. So to like validate this again, we kind of like use like a protein, a different kind of like a different assay. So we're doing immunohistochemistry of MVP on tissue. So we're looking at gray matter and also white matter. In E2, it's maintained. In E3, we see a reduction of MVP. So I guess you can see that this is lighter. But then interestingly and kind of surprisingly, you actually see an increase of MBP and AD. Overall, there's just lower MBP levels overall, but like the this increase is, I guess, unexpected, but it's also been seen elsewhere. So kind of the thought there is like, so this is like a little counterintuitive, right? Like if MVP is like, if it like, preserving that like myelin sheath and like protecting the neurons, like why is it in E4 where we see like worse cognitive outcomes? And kind of like the thought is that it might actually be like degraded MVP. So yeah, that myelin damage in AD leads to an increase in DMVP, which like might be why we see this kind of like counterintuitive result when we look at MBP. So yeah, so if that MBP decrease, like, is this preserved MBP, which would kind of be counter to like what we see in like cognitive and health outcomes, or is this preserved and like, or is this degraded MBP? So they did another assay looking specifically for degraded MBP. In ApoE, we see minimal degraded MBP. In ApoE4, we see really high degraded MBP. So that's kind of supporting like that's what's actually happening in this change. So overall, kind of thinking about like those three sets of results together, the transcriptional changes, the immunohistochemistry with MBP, and then like looking specifically at the degraded MBP, kind of the conclusions we can draw is that like E2 is helping preserve the myelin content. We're seeing like high levels of MBP. It's preserving it across control and AD cases. E3, we see MBP loss in AD. And then E4 carriers, where there seems to like the amount of MBP seems to be preserved, but it does seem to be damaged. And we see an accumulation of damaged MBP. And then, okay, so then turning our attention to microglia, which is the cell type implicated in AD. So for ApoE4, we see an enrichment of inflammatory responses in microglia. They subcluster their microglia to 8 microglia and macrophages. Again, we don't find any differences in those fine cell type proportions. They find the inflammatory and surveillant microglia populations are enriched for inflammation response between AD and ApoE, between AD and control. And then they find that homeostatic microglia had distinct changes across their genotypes. And then I also noted that their annotations, because they kind of like drew on two other papers, the Sun et al. And the Printer et al. For marker genes and these annotations, that these could be helpful for our study where we're looking at microglia subpopulations as well. For differential expression, ApoE3, we did not see or had the most differential expression signal. ApoE4 had the most genes not differentially expressed. So we see not a ton of differential expression signal in E4 between case and control. So their kind of thought there is that like, are the microglia just not responding to alpha beta plaques in the brain? Like here that like we see like differential expression between case and control, the microglia are adequately responding to like alpha beta acclimation, but in E4 like we don't see that change. So are they just like are they not doing their job there? So then they do another assay where they're investigating that. So I think that they study specifically like so yeah, looking at like the specific like uniquely differentially expressed genes by group. And then also, if I remember what assay they did here, they have confocal images of DM. Oh wait, sorry, that's the last one. Yeah, we have confocal images looking at CD68 and IBA1, which I believe are markers for microglia in like the AD. I think like you can see like we see more microglia around an alpha beta plaque, more microglia around alpha beta plaque, and then in E4 you see less red, which is the microglia. So kind of like confirming that hypothesis. And then so like yeah, these ApoE4 might mediated that microglia response and like they might not be responding adequately to the alpha beta plaques. So I guess excitatory oligos and microglia were like the major populations that they like really delved into and kind of those major responses. But they also like note later in the paper, and they have a lot of supplementary figures as well, changes in other cell types. So inhibitory neurons, they not have similar transcriptomic changes as excitatory neurons, and they find the most differentially expressed genes in E2. OPCs, they found changes in the immune response pathways, and there were some differences between subclusters. For astrocytes, in E3, E3, they find distinct differences. They find an increase in cytokine production, and then in vascular cells, they find changes in immune response in E2, cells. They have this little graphical summary that like breaks down some of these differences. So then moving to their discussion, they point out that some weaknesses that they had in this study were low sample sizes. So like they only have like four, broken down by genotype, sex, case, and control. They have about 4 or 5 samples there, which is pretty decent, but like there could always be better power. And they said this specifically that this would help them find more differences in minor cell populations. They also note that they found those discrepancies between transcripts and protein changes. So I guess like going back to like the like MVP where like when they actually looked at the protein changes in E4, like it was different than they expected from the transcriptomic data. So that might not necessarily be like a problem with their study, but rather like the like steps in between like the RNA and the actual proteins. Something's happening there that needs to be studied that they're just not finding here and don't have the resources for. But overall, their main findings like did fit mostly within like with like other knowledge and like support other findings in the field. Noting synaptic dysfunction, where E2 is protective and E4 is damaging, specifically in like neurons. Also like the myelin damage, where E2 is protective versus E3. And then kind of this interesting note where like E4 has an increase in like AD increased MVP, but like it's like a degraded MVP. And then microglia, finding those upregulations of those pro-inflammatory pathways and like perhaps this like inadequate response in E4 microglia to alpha beta, kind of like again, kind of like making sense of that maybe counterintuitive result they got. But and then like differences in the microglia in this study did differ from studies with like animal models, but they note that like this is probably real and like just because like these are human microglia versus like mouse microglia. So obviously the mouse models of like any brain disorders are pretty tricky. There's big differences between the mouse brain and human brain. So this is always kind of an issue in the field. But yeah, overall, many complex changes across cell types in AD modulated by ApoE. So there's a lot of different axes of change between cell type, between AD and control, and across ApoE. So it was a ton of data to crunch, and I think overall they had some pretty interesting findings. Cool. With that, any questions?